Alrighty, good morning. Oops. Today we're going to go through the cell wall structure. So we're going to kind of break down the cell wall, talk about a little bit more about what it does, but mostly how it's set up. So this will be pretty crazy. So my guess is you've never looked at the cell wall this closely before. I know when I learned this, I didn't realize how many parts there were to the cell wall. So first thing though that I want to hit is just to kind of a review over what cytoplasmic streaming or cyclosis is. So it's just to remember that we talked about it on the last lecture with mitosis and cytokinesis, but it's going to help facilitate the exchange of materials within the cell and between its environment, so inside and outside. When we get towards the end of this part of the lecture, we'll talk about specifically where in the cell membrane the exchange of materials occurs. So, and then just so you're aware, chloroplasts will move in a counterclockwise fashion during the cytoplasmic streaming, just kind of around the outside edges of the cell because that's where they're located. And also, if you recall, the vacuole makes up most of the inside of the internal volume of the cell, so really the only place for the other organelles is kind of on the side. So, all right. So a couple of characteristics of the cell wall. It's one of the primary features of your plant cell. It's going to be very rigid. I believe what I've told you before is it's there mostly for support and structure, which is true. But specifically, we're going to go a little bit more in depth. The thickness of your cell wall is going to depend on what your cell is doing and where it's located. Sometimes you need a thicker cell wall. Sometimes you need a thinner cell wall. Depending mostly on if it's there mostly for transporting things inside and out, or a cell that needs to transport things in and out of it, or if it's there mostly as a support and structure type of a cell. A couple characteristics of your cell wall. It does help limit the size of your protoplast, and if you recall, the protoplast is just that plant cell without the cell wall. Another thing that it's going to do is it's going to prevent the protoplast from rupturing. So, which is obviously a good thing. You don't want the cell to explode. It's also going to, like we've said, it helps serve as support and structure for your protoplast. Yes, the cytoskeleton aids with that, but this serves as more of external support rather than internal support. That's really what the cytoskeleton is for the internal support. This is more for the outside. And then at the very, very end, we'll talk a little bit more about this, but it does allow for some transportation, secretion of hormones or other chemicals, and it allows for some absorption. With some bacterial cells, I'm trying to think here because it's been a while, I just thought of this. With some bacterial cells, they will pick up certain dyes and pigments, and that is how they get classified. So, yeah, that's kind of what the absorption for the cell wall is. So, okay, so this is a little smaller, so I'll try to. It's not going to let me get it too much bigger, unfortunately. So, okay. Yeah, still not going to let. Okay, sorry, I'm trying to make it bigger for you, and it's not working so well. So, hopefully you can see that semi-well, but okay, we're going to talk a little bit now about the components of the actual cell wall. So the primary excuse me, the primary component of your cell wall is cellulose. But I believe we've talked about that, so I think you know that you're familiar with that. If you remember from last semester, cellulose is a polysaccharide, which means it's a sugar or it's a carbohydrate. So it's going to be made up of those repeating monomer units of glucose in this case. But this is, excuse me, this is a primary component. So another thing is hemicellulose. So another polysaccharide, but its job is to regulate the cell enlargement. We've also got pectin, which we talked about last time. It was produced by the Golgi, those pectin-filled vesicles, during cytokinesis. So, and then when those vesicles released or burst, they released the pectin in between the two new cells. And remember, it's kind of the glue. So, so it's just found in between. It's primarily going to be found in between adjacent cells. That's going to be where it is. Now, if there's cells all the way around your cell, obviously then the pectin would be spread throughout, maybe not on the inside part, the inner cell wall. So, yeah. It is hydrophilic in nature, so water soluble. But with the next one is lignin. With lignin, you have I'll talk about that later. Never mind. Sorry. I've got like 60 things in front of me here. The lignin. This is going to be put in your cell wall for reinforcement and strength. So kind of fibers there to help with the actual structure of the cell wall, which helps with cell structure. It is hydrophobic in nature, so not water soluble. There is some water inside your cell wall. It's going to be mostly found in the primary cell wall. That one with, I don't know how well you can see it, it's a one with a little degrees sign in the superscript. That denotes primary. I'm sure you're aware of that. I just don't know how well you can see it on the screen, but you've got a primary and a secondary cell wall, so the water is really only going to be located in the primary cell wall. I have here that it's about 65% water, the primary cell wall, when it's growing. So, and then the last thing, you've got a few other smaller bits and pieces that make up your cell wall, which would be various proteins and waxes. These are going to be found in the outer protective tissues of your plant, and it's there, waxes especially are there to help prevent water loss. Makes sense. If you look at the top of your diagrams this time around, I've drawn you kind of a doodle of two plant cells just to kind of reacclimate you with the structure of your basic cell, your plant cell. You've got your protoplast there in the center. You've got your cellular plasma membrane there as the inside line. I didn't label that. And then the outside line would obviously be the cell wall. You've got your middle lamella there in the middle that was formed when when cytokinesis occurred. So, and then you've got those pieces of endoplasmic reticulum in the middle of that middle lamella, and those are the plasmodesmata, and those are those communication channels in between the two cells so they can communicate between one another. And the middle lamella, if you remember, that's what's made of the pectin or the glue. That's the glue that's kind of holding everybody together. So, all right. The next part is the structure of the cell wall. So, talked a little bit about what it's made up of. Now we're going to, how is it all put together? Okay, that first line, the second set of doodles there where I have glucose, etc., makes up your cellulose. That right there, I think actually I'm jumping ahead of myself, but that's actually going to be how your cellulose is made. So, and yes, I did jump ahead. So push pause on that really quick. Sorry. So your protoplast is going to make your cell wall. Your cell wall consists of three basic things. You've got your primary cell wall, your secondary cell wall, and your middle lamella. There's not much to say about the primary cell wall other than it's thin compared to your secondary cell wall. It's relatively thin. I believe it's still thicker than your plasma membrane, but I'm not sure by how much. Your secondary cell wall is much, much thicker, and there's reasons for that, but it's there more for the support. You could argue your primary cell wall is there more for the protection part. Since it's the furthest out, it's going to be the furthest out. So the way this works, and you can kind of see it's on the back of the I guess it's the front side of the second page. Sorry. You can kind of see your secondary cell wall is going to be cozied up right next to the protoplast itself. And so the primary cell wall is made first. Then the cell starts producing the secondary cell wall, or starts putting together, rather, the secondary cell wall. So it's inserted in between your primary cell wall and the protoplast, which would be technically the plasma membrane. So that's how it's formed. So protoplast has the plasma membrane. We produce the primary cell wall, and then when we're done making the primary cell wall, we're going to insert the secondary cell wall in between the primary cell wall and the plasma membrane. So you can kind of see that it's the page number is page 21. It's that top drawing. You can kind of see the order of the cell walls. And then we've got the middle lamella, which we talked about, which was the glue or the cellular cement. We just talked about how it was made of pectin. It actually can have it attracts ions, so you can have calcium pectin or magnesium pectin. It's just different types of pectin based on what the cell needs. So just so you're aware of that part. Okay, this is where the diagrams, this is where I was ahead of myself, sorry. So now go back to the drawings that I drew you. So now we're going to take the primary and secondary cell walls and make them even smaller. We're going to break them down into their individual components. The smallest, sorry, still at school. So the smallest part is the individual glucose molecules, because your primary component of your cell wall is the cellulose polysaccharide. Break that down into the individual monomers, which would be your glucose. So repeating units of your glucose make up your cellulose strand. And then if you look at that cylinder-looking structure there that I drew, I've taken different cellulose strands, which is the G plus G plus G. Each one of those is a cellulose strand. If you get 3 strands of cellulose, that's what's called a micelle. Okay? So cellulose makes up a micelle. Then what you're going to do is you're going to put strands of the micelles and then that's what that bottom cylinder looking strand is. It's a bunch of my cells, or layers of my cells, and that's going to be called a cellulose microfibril. You could just call it a microfibril, but because it's made of cellulose, we call it a cellulose microfibril. This is going to be found in both your primary and secondary cell wall. So it's going to be the same basic structure of these microfibrils. Now, how the microfibrils are oriented, that's what's different between the primary and secondary. Okay? If you go down to the next set of doodles on the very bottom where it says matrix construction, your cell wall is considered a matrix because it's got multiple parts, but your primary cell wall has a very random arrangement of microfibrils. They're not ordered, they're just randomly inserted, and that's kind of what I've attempted to draw. So those are those haphazard arrangements. Your secondary cell wall has a very specific orientation, and it kind of looks like I can't think of the word now, but the zigzag pattern. So there's 3 layers within your secondary cell wall. You have an S1 layer, an S2 layer, and an S3 layer. Not sure how clear that's coming up on your screen, but that's what those little subscript numbers are. It's S1, S2, and S3, and it goes in that order from S1 being the furthest out, closest to the primary cell wall, and then the S3 layer being closest to the protoplast, so right up next to the plasma membrane. So they are oriented in that fashion, in that zigzag pattern. It's not as apparent in the primary cell wall, but you can definitely see it in between the microfibrils in the secondary. You can see the little squiggle lines in between each microfibril, and I tried to draw them up in the primary. It's just harder to see because of the crazy orientation, but those little squiggles, that's going to be your hemocellulose and some of those proteins, those other components of your cell wall. So your lignin, the waxes, all that stuff, that's all going to be kind of in between, in the secondary cell wall, in between the 2 microfibrils, and then it's just going to be embedded throughout in the primary cell wall there. So that's how your primary and secondary cell wall, if you could get a microscope that was powerful enough to be able to go that small, that's basically what you would see. So random orientation of your microfibrils in the primary cell wall, but the layered orientation in the secondary cell wall is kind of what I want you to walk away with that one. So, okay. Your construction of your cell wall. Gosh, that's tiny. I hope you can't read this, email me and I'll send you the PowerPoint. So I'm using the Chromebook, so I'm using PowerPoint online, so it's not the best. Okay, so overall, I kind of referred to this earlier because the title of that last set of doodles was the matrix construction. Like I said, it's a matrix because it's a bunch of different parts kind of put together. So they tend to say that the construction of this matrix, your cellulose and your proteins and your lignin and pectin and all that, is kind of like a sidewalk. Think of it kind of like a sidewalk or a crossroad or something that uses cement with rods. So normally before they pour that cement, they put rods through it to provide some additional support and structure, and then they fill in with the cement that eventually hardens. So it's kind of that. We've got the rods of cellulose, microfibrils, and then the cement would be your pectin and your middle lamella and stuff like that. So, okay, if you look on the back side of the doodles, it's I would call it the second set of doodles down on the left side. There is a little your plant cells aren't exactly rectangles and squares like we've always drawn them for you in biology. Oftentimes they're kind of hexagonal, or they're still usually very they're not like circles. They still have points, but they're not a perfect rectangle or square, and not everything lines up perfectly. So oftentimes there's these gaps in between your cells, and that is called intracellular spacing. And that diagram kind of shows you where the intracellular spacing is. This is really important for the plant tissue as a whole. This is where some gases are stored, so that's where the gas exchange is able to happen. So they might store excess oxygen or carbon dioxide or something there in those middle spaces. So the extracellular spacing is important. While we're here, I'll kind of talk about those two protoplasts up above on the very top there. The protoplast on the left only has that primary cell wall, and then the inside ring which isn't labeled, that's your cell membrane or your plasma membrane. The larger cell there to the right of it, that's got both the primary and secondary cell wall with the plasma membrane, and then the protoplast. So, alrighty, last part that we're going to cover for today is, talking about cavities or thin areas in your cell wall. So oftentimes these are necessary to exchange fluids or sometimes other necessary molecules for the plant cell. You can kind of think of them like pores, but we call them pits. So these are indentions would be cavities. So oftentimes they're going to be at a thinner area of the cell, which makes sense. Why would you want to carve out a cavity in a thicker area? So usually they're going to take place they're going to be found in the thinner areas of the cell wall. And like I said, it's usually there for the exchanging of fluids. So you've got pits and pit fields, which I'll talk about here in just a second. So, all right. Two places that you can have pits. You can have them in the primary cell wall if you just have a primary cell wall, or if you have a secondary cell wall, you would have pits. So in a primary cell wall, it's called a pit field, but if it's located in the secondary cell wall, it's going to be called a pit. So I'll be flipping back and forth between these notes and the diagrams. So on the back side of that first page, so you pretty much tell because it's got pit fields and pit membranes all labeled. So those are going to be the diagrams we're going to reference here now. So the first one that's next to, so it's the second one on the right side. It's across from the intercellular spacing diagram. I went ahead and outlined the cell wall for you, just visually so I could see it. So, in like I said, if you only got the primary cell wall, the indention or that cavity is going to be called a primary pit field. So I've labeled that on both cell 1 and cell 2. It's those little kind of indentions or the cavities. You've got your middle lamella there running in between that's holding the cell 1 and cell 2 together, and then the plasmodesmata there is in the center with the lines going across there. So they still have the plasmodesmata there. It's called the pit field. If you have a secondary cell wall, because some plants do not have a secondary cell wall. Most of them do, I believe, but not. There are some instances where you only have one. You then have what are called pits. Okay, and they can be simple or bordered depending on their shape. The left side is going to be the simple pit, and then the right side would be the bordered pit. So depending on which cell type or which tissue type you are, that's going to determine whether or not the secondary cell wall is going to become a simple pit or whether it's going to become a bordered pit. So the next lecture that we'll talk about is like some of the different cell types that you can find in plant cells. And so, for example, if you're a parenchyma cell or a collenchyma cell, that might determine whether or not you're going to become a simple or a bordered pit. Okay. A simple pit is very open. I'm going to say is very open. This particular one is drawn kind of like a rectangle, but I've seen them drawn also as a circle. The difference is between the simple pit and then the bordered pit. If you look at the bordered pit next to it, the bordered pit, it's almost sunk in, if you will. So the secondary cell wall kind of comes around it, so it appears more closed off. And then the simple pit, it's very open. I hope that makes sense. If it doesn't, let me know. But that simple pit is very open. The secondary cell wall does not come back in on it to kind of close that opening there. So the two pits right next to each other are going to be what's called a pit pair. And I've labeled that on the far left. So when you've got the two pairs next to each other, that's called a pit pair. So you can have a pit pair with a simple pit or simple pits or with bordered pits. So, but like I said, these are important so that way some of the fluids can be exchanged between the two cells. So on the back side, or I guess the next page, 21, which would be the front side of that third page, the second page, I guess, sorry. It's technically the third page, but it's the front of the second. We've kind of already talked. This is a nice cleaned up version on top of the cells, so you can see that this particular cell has a pair of simple pits because they're open. If you've got the bottom, excuse me, the bottom part of the diagram there is just talking about if you've got the cell, the secondary cell wall, that primary cell wall is very, very thin. So it's just kind of showing you just a zoomed in look there. So, but that's what's called the pit membrane. I think I labeled it on page 20. So that's just where the two primary cell walls hook up with each other. It's the pit membrane. So you have one of those when you've got a secondary cell wall. Technically don't have that with a primary cell wall only. It's just called a primary pit field. So like I said, this it's a little more in depth probably than you've ever gone into the cell wall before, so hopefully it makes a little bit of sense. And I believe you've got a worksheet that kind of goes over mitosis and cytokinesis and everything. So if you do have any questions, please don't hesitate to ask. And tomorrow we'll talk about the different cell types found in plant cells. So it'll be great fun. So I guess that's it. So have a good day.